recording, and I think we're recording now. So, first of all, thank you for joining me tonight. And I was going to do today just a really quick kind of a review of where we are and what this week's material contains, and then go through the material of the urinary system. But if at any stage you have questions, you just shout out to me. And I think perfect. I think that this slide is a kind of a realistic of where we are standing to remind us that we were planning to be on week 12 on the next chapter, which is all about the fluid balance, electrolytes, pH balance, and so on. But obviously, we're now working on the urinary system. So we're working on the material from week 11 on a week 12. And we'll talk about what that means for this class. Just briefly, I kind of have addressed it earlier on, but I want to make sure that I'm clear about that. So what I'm gonna spend the initial moments of our time together is just to go through the Maricopa systems outage, how that affected us, how we are doing with the exam and practical too, and if there's anything else that's important. But the main focus of this session is going to be about the assignments for this week, how the lab, which is a little different, how that works, and then really focusing on the urinary system and reviewing the material from that. So nothing too surprising from the format that we've used before. So I think I used these slides last week on my session. So basically, just to kind of quickly remind that and provide you the most recent update. So Maricopa Systems, which covers multiple colleges, we had an IT outage. I believe it started on the 16th. And as a result of that, there's been plans in place for this already ahead, that if there's anything that doesn't seem to be working the way it's planned to work, let's take everything down and then bring them up, just to be sure that safety and security protocols are followed and no students' information is at risk, no staff members, faculty members, administration members information is at risk. So that was done, and the goal was always to resolve it quickly, but they just wanted to play it safe. So for that reason, the spring break was extended by 1 week, and it will have few effects on our class. But this update that I have up there for Tuesday, that's week ago Tuesday. So already then all of the operating systems were up and running. So email, Google, tools that we use, employee and student portal, and internet. And as you may have noticed during that spring break and that extended week, when we were still not resuming to the classes, all of the staff, all of the faculty, all of the administration, as well as students, had restricted access to our tools. So for example, I didn't get access to our emails or to our Canvas or other places. So for that reason, if you had to wait for an answer from me, that was the reason. But I think I should be pretty well up to date on all of the messages. I think there's a couple of new messages on the Canvas, and I'll address those as soon as I can. And the classes resumed last week. So last week we had a look on this class of the respiratory system. And this week we're going to have a look of the urinary system. But the big thing to remember on all of this is that both college and me as well, I always say this to you guys, have made a commitment that under no circumstances are your grades going to be impacted because of this whole thing that happened. So what's happening instead is that the last day of the semester was pushed back by a week. So that means that whereas we were supposed to finish on one date earlier on, this course will run for another week after that. I was saying earlier on, just before I turned on the recording, that I do realize that some students may have other plans. Students may have, for example, planned to move on to work immediately, or students may have planned to travel. So I do realize that that might be the situation. And if any student wants to submit work ahead of the time, I will work with you to make that happen. However, what's lucky for our class is that I always keep my finals week as a project that you guys do. So basically, it's just doing on a computer like a virtual poster, and all of the points from that activity are extra credit towards your final grade. So in a way, you're not going to lose out on anything if you choose not to do it, other than just getting those extra credit points. So we will continue 1 week longer, but if you cannot do that, that's absolutely fine. And you can even do the finals week assignment 1 week earlier. So there's no issue on that sense. And as you may have noticed, all of the due dates as well as the chapter materials, they were pushed forward by 1 week. So we're covering this week material that was initially scheduled for last week, but also all of the deadlines that were initially last week are now going to be only at the end of this week. So there's no big changes, nothing to worry. And there's a couple of things that I'm still working out what's the best way to do in terms of if someone has to finish the semester a week earlier than we actually finish. But I'm going to share information about that in a due course. But really, nothing to worry. We just proceed based on the schedule that's on your Canvas on those learning modules. So I changed all of those. What I did not do, I did not go back to the syllabus because I thought that that might be just too much confusion and trigger a chain reaction of other things. And on the syllabus, I have the schedule what we're covering each week. So if you're following that, just look back 1 week from where the date is. And that statement is at the top of the syllabus that I did change. In terms of the exam and practical too, obviously because we were all locked out, I was locked out as well, and I couldn't grade those, but I have them graded. There's just few little details from few students that I want to check. I was hoping to have those released today. I think it's going to be more likely tomorrow, but either way, I will post an announcement when they are ready, when they are finished, and when you can expect to be able to see your final grade for those. But why I wanted to take a time to mention this is that I have had some students being really concerned and sent me a message that, oh my God, I thought that I did better on the exam too, or I did better on the practical too. The score that you see now is the score that the computer, when it automatically grades those, what it gives. And we all know that what it gives for you automatically is not the right score because you normally actually scored way better than what it gives you automatically, because the computer software behind that is very simple, very stupid. So it doesn't account spelling errors. It doesn't account for partial points. It doesn't account for many other factors. So most likely on average, just kind of an average feeling, normally my students have their grades go up about 10 points or so at the minimum. But that varies, of course. If you didn't answer anything, it's really hard for me to give them points. But don't panic with whatever the score you have for the exam 2, practical 2. At this point, that score is not the final score. Your final score is probably going to be better. That was a very long-winded way of explaining it. The other thing that I did want to mention is that if you have any assignments that you missed out, didn't get a chance to answer at all, or you didn't do as well as you wanted, and you're looking back and like, ooh, I could have done better on that. Or if you have an assignment that you're running out of time to get it finished, you can always message me. I'm super happy to reopen those, extend due dates. All I ask is that just be very clear, give me specific that what assignment it is, so I can then find it easily. And normally the quickest way to reach me is through Canvas. I, of course, check my emails as well, but there's a couple of messages, but I will definitely get back to those. But normally that's the quick way to get hold of me. If you don't hear back from me and you're getting a little nervous, just resend that message again. I will, then it jumps on top of my message list. I will answer. There's sometimes things slide through the cracks and I won't see them anymore if I've read them but haven't answered or haven't acted. So I don't feel bad at all about resending anything. What I wanted to speak about this week's assignments is that all of the assignments that we do this week are completed through the Connect from the publisher's website, McGraw-Hill's website, and they are all familiar to what you've seen before. We have the Learn Smart, we have the quiz, and we have the practice anatomy. They all are due at the end of this week, I think 1 minute before midnight. The only difference, and I said the difference this morning, but after that I started thinking that actually I think we've done this once before. But in case someone missed it, I'm going to talk about it real quickly once more, is that our lab for this week is going to be one of those Phil's labs. And what it means that it is a Phil's lab, that means that that lab requires you to have a flash on your computer. So you will need to be able to run a Flash application so you're able to work through that lab. And the reason why that might be a challenge for some is that I believe the official support for the Flash ended at the end of last year. So newer browsers are already blocking the Flash, that they're not allowing the Flash to run automatically. So you might need to do some little extra steps to get it to work. You will notice that the lab that I'm talking about is labeled as ADH lab stimulation. And actually, I believe it's the very first activity on this list of the assignments that I have here. I moved it around from here to here so that it would be very, very obvious and very easy to find. But anyways, if you're struggling to get it run, I have here excellent instructions on how to get it to run. And if it still doesn't work, I will be super happy to help you. And if it still doesn't work, then I will come up with an alternative lab that you can complete. But let's have a look at what happens with the Flash. So you will need to, if it doesn't run the lab, you will most likely need to either install a Flash player or what's more likely scenario, enable it, so allow it to run. If you don't have a Flash player at all, you will get some sort of a notice, as you see here, that, oh, you need to have a Flash player, please download it here. If that notice doesn't come up, you likely have the Flash player, but it's been just blocked. So what you will see is at the end of this address bar on your web browser, where you would normally write the web address where you go, at the very end of it, on the right-hand end, you will probably see a symbol that looks something like a lock and a slash through it, or a red line through it. And you click on that symbol and you choose Allow Flash. This is what most students do, and it runs nicely. There are students who it doesn't still run. So in addition to doing the step A, you might need to do also the step B. And in that case, you look to the other end of the address bar, to the left-hand end, and you will see something like an exclamation mark or a little text that says that this is not a secure website or secure activity. And you go and click on that and allow the Flash to run. If you click on that, what you will see, for example, for me, it had an exclamation mark and a text not secure. I click on that, it opens this nice page or nice drop-down menu, and I look for the Flash. And from there, from the drop-down menu, you choose Allow. And if you do both of these, the lab will more than likely run. So where you will end up is a site that looks something like this. It has the fields mentioned at the left top hand corner, and it has a menu of all the labs that you can choose from. And what I'm asking you to choose is to choose the lab number 42, which is all about ADH, so antidiuretic hormone. And then you just work through that and you're set. So if you get stuck McGraw-Hill, our publisher, also has IT technology support number where you can phone, but you can also message to me. We can do face-to-face or kind of a virtual face-to-face meeting and figure it out together. And even if it doesn't still work, well, then I can give you an alternative lab that you complete instead. So don't spend a huge amount of time fighting to get that lab to work. What I want you to do is focus on the actual work, not on how to get it to work. So that's all I have to say about the Fields Lab. And I do have a feeling that we've used that before. So it's exactly the similar to that. And I have a feeling we used it for doing ECG or something like that. What I wanted to do with the next section was to go through the main points from our chapter on the urinary system and what's important on that. What you'll notice when we go through this chapter is that we spend most of our time talking about one single structure. And the structure that we will be looking at is going to be nephron. A nephron is the structural and also functional key unit of the urinary system. So that's why we spend a lot of time on that. We will also cover the basics, the kidneys, ureters, urinary bladder, and urethra, but it is the nephron that we will be focusing. So I'm going to jump out of this presentation, and what we'll do, I'll open the presentation so we can have a look of what's the key points from this chapter. As always, if you have any questions, you can just message me and or turn on your mic and speak to me. I don't want you to have to wait to the very end. I want to answer things immediately. If you find that you learn well by taking notes, we will be drawing the nephron together. So you might want to have some sort of a piece of paper where you write and maybe a pen or colored pencils, whatever works for you. But I don't want you to feel that that's the only way how you can learn it. You do what works for you. I'm not expecting you to do, if it's not the way you learn, you don't have to do that by any means. But that's something that we will be doing today. We'll be drawing it together. I find that most students learn that way the best. So here we are on our chapter of the urinary system. And like I said, it is going to be the nephron that will be focusing largely on this system. But I want to start with this formula or a chemical series of reactions, which is now scribbled all over, but we can use it regardless. And we saw this series of chemical reactions at the end of when we talked about the respiratory system. But what I'm wanting to show with this is really, and don't think that you have to learn it by heart. You don't have to learn any of those symbols. You don't have to learn the names of the symbols. This is just me trying to show that all of these systems are connected. So I'll talk a little bit just to get us thinking is that how the digestive system, respiratory system, urinary system, and actually also cardiovascular system all are connected to each other. So what we see here is very basically that you get from your diet, from the food that you eat, various different kinds of chemicals, and some of those chemicals we can use for energy. And most commonly, that energy that we can get from the food is in forms of sugars. So what we have here is a sugar molecule or sugar molecules if we added the balanced equation. In addition to that, you will need to get some oxygen, and we get that oxygen from the respiratory system. So by breathing in that and then using the cardiovascular system to transport that oxygen to the tissues, where all of the next steps that we're going to have a look of are going to take place. So what your body does with sugars and oxygen is that it turns it into ATP, which was the chemical energy currency that your body uses. And ATP is really what enables your body to do all other things. that it does. It's the energy, the driving force for all of those. But as a waste product from that process, we are left with carbon dioxide and some water. And this is where this carbonic acid equation that we see on the bottom comes in handy. So we need to find a way to get rid of the carbon dioxide and water. What happens is that gets turned into carbonic acid, which we are able to transport in the blood. And then that carbonic acid can be broken into bicarbonate, and we can actually breathe out the bicarbonate by a respiratory system. So we transport the bicarb to the lungs and breathe it out, but we're still left with these extra hydrogen ions. And how we get rid of the hydrogen is largely through urination. So we will be looking at the urinary system today, and how it all ties into this is that the urinary system is what we use to get rid of these hydrogen ions. But like I said, this is not a formula that I'm expecting you to memorize. It might be something that you recognize from your BIO201 or BIO156 or whatever general biology class you've taken in the past. But simply what I want to show is that we need all of these. We need the digestive system, we need the respiratory system, we need the urinary system, and we need the cardiovascular system to work together. If we take away one of these systems, none of the other systems are able to complete all of their functions successfully. But what we're going to do, what we are going to focus on, is that we're going to talk about the urinary system. And we're going to spend the majority of the time looking at kidneys. And one of the things when I ask from my students that why do we have kidneys? What do kidneys do? Most often they answer to me that, oh, kidneys are what we use to make urine. So we use kidneys to make pee. And that is true, but that is not really the key function or main role of why we have kidneys. Yes, we make urine, but the key function, the main role of the kidneys is that we use them to filter blood. And while we're filtering blood, we're able to monitor the blood pressure and do adjustments. If the blood pressure is too high, we can then get rid of some of the extra fluid in there and urinate that out. If the blood pressure is too low, we can stop the urination from happening and let the liquid levels build up. So kidneys play main role in the regulation of your blood pressure through filtering the blood. And of course, as a byproduct of that, we do produce urine. There's a bunch of other functions as well that are important. We will, on this session, quickly look at the ureters, urinary bladder, and urethra. Notice that we've got 2 kidneys, 2 ureters, 1 urinary bladder, and 1 urethra. But we'll go through all of those one by one. What I have here is some of the functions that kidneys have. And of course, there's a lot. They do a bunch of things that are important for us, but we're going to focus on this filtering blood and how filtering blood helps us to regulate the blood pressure. Well, what we see here, we're going to start by talking about the kidneys on terms of their anatomy and looking from external level. So I have already colored this. This is from the morning session, so I didn't remove the colorings. We'll just work through them together. So I'll just take a pointer instead of a pen. So if we look at a kidney and start describing what kind of an organ kidney is, a single kidney is usually about 12 centimeters long, about 6 centimeters wide, and about 3 centimeters thick. So not huge, but still decent sized of an organ. They are usually reddish brown in color and kind of a bean shaped, if you wish to describe that with some sort of a shape. Another thing that we notice is that they're really smooth from the surface often, and this is because kidneys are enclosed inside this fibrous capsule that is known as renal capsule. So that's why they seem so shiny and have such a smooth surface. But what I wanted to do is look at the kidney on an external anatomy here. So let's talk about that. And we're going to start by the fact that the medial surface, so this surface that's facing to the midline of the body, so on both of these sides facing to the middle, the medial surface of a kidney is always concaved. If you look at it, it has this curvature that's curving so that it's opening to the inside, to the middle of your body. And that's going to be important for a few structures that we will notice on this concave surface. First of all, we will notice that there's multiple blood vessels coming in. There's also lymphatic vessels, and then there's nerves. So a lot of things going into the kidney and a lot of things also leaving the kidney. So clearly there's happening a lot. And if we remember the main function of the kidney being filtering of the blood, so of course we want to get a lot of blood there. And we're also going to take away a lot of blood. The lymphatic system was the complementary system to the cardiovascular system. So it makes sense that we have some lymphatic vessels going there as well, or picking up the lymph from there. And of course, some nerves, because it is a highly active part of our body, and we want to be able to monitor that what's happening there. Well, the other surface of the kidney that we see on this lateral side, so facing towards the sides of the body, that's going to be convex. And on this convex surface, really what we're seeing is that it's much larger. And when we are starting to look at the internal anatomy of the kidney, we start to see a lot of interesting facts. So why don't we head towards having a look of the internal anatomy of the kidney? But just before we do that, there's a couple of things that I still want to highlight from this external part. First of all, kidneys are located behind the peritoneum. That was the layer that we saw lining the abdominal cavity. So for that reason, we call them retroperitoneal, so behind the peritoneum. Another thing that we notice is that our left kidney is located higher than our right kidney. And the height difference, that can vary a little bit, but there is a little bit of a difference. So now the good question would be that why is our right kidney lower? What's making it be so much lower than the left kidney? Well, the explanation to that actually will become obvious when we think of the other abdominal organs that we have on this area. And I know that we haven't yet covered the digestive system, but this will become more obvious when we talk more about digestive system. Well, we have liver at the very top part of the abdominal cavity. So liver is this kind of a wiggly shaped organ that's larger on the right-hand side of the abdominal cavity, and only very tip of it points to the left-hand side. So because the liver is immediately on top of the kidneys, it's taking most of the space there, and that causes that this right side of the body in terms of the kidney is kind of pushed downwards. So that's the reason why the right kidney is located lower, simply because liver takes most of the space on top of that. Because there's no liver on top of the left kidney, or there's less liver, it's able to locate higher up. And finally, one more thing that we see when we look at the surface anatomy, external anatomy of the kidneys, of course, our good old friend, the adrenal gland. We went through the endocrine system and saw that adrenal gland was this gland that was located on top of the kidneys. So we recognize that structure there as well. We will not go into great amount of detail on this session about clinical issues that can happen, but I do want to mention that there are various things that can happen, and the reason why we have to learn about the normal anatomy and physiology is that so that when you move on later in your studies to learn about all the pathologies, you need to know what's normal before you can learn about what's abnormal. But one abnormal example that I've highlighted here is hyponephrosis, where there is urine that's backing up. It's not flowing away like it should. And if it's not flowing away, more urine is still regardless going to be done. So if there's a blockage somewhere, that can end up on buildup of this urine, backing it up, and eventually end up damaging the kidney. So that would be a problem. So this is really the reason why we want to go through the normal functions before we have a look of those in your later studies. Well, let's have a look at the internal structure of the kidneys. First of all, what we see here on this kind of a turquoise color, the outermost, the superficial layer of the kidney is known as the cortex. So just like you had a cortex for the brain, the outermost layer of the brain was this gray in color, and that was our cortex. Similar with the kidneys, you have the outermost layer, which is cortex. In this case, though, it is paler in color than the other parts of the kidney. So in addition to having this cortex, we of course are going to have medulla. So medulla is all these parts that are deep to the cortex. So below, underneath that, when you dig into the kidney and look inside there. And one part of the renal medulla that we see are these pyramid-shaped or cone-shaped structures, and they are known as renal pyramids, or sometimes medullary pyramids, or sometimes medullary renal pyramids. But they are renal pyramids because they look a little bit like pyramids, being this kind of a triangular in shape. They're usually darker tissue. The other structure that I want to highlight here is this kind of a funnel-shaped tube that starts inside the kidney, and then as it leaves the kidney, turns into a ureter. This part that's still within the kidney is known as renal pelvis. So those are some of the very good parts of the internal anatomy of the kidney to know. But like I promised to you, we will spend most of our time on this session talking about the nephrons. So nephrons are both the structural but also the functional unit of the kidneys and of the urinary system that really is the business end where everything that's important for us in a big scale happens. Other things then just help with whatever follows afterwards. So here I have drawn a diagram of a nephron. And that diagram, I think it's pretty good. Your textbook uses a little different kind of diagram. But what we're going to do, we're going to draw a nephron together. Because I find that most of the time, my students learn the best when they do it themselves. So I'm going to draw a nephron with you. If you do not learn that way, if that's not helping you at all, you can simply watch. Or you can simply listen, whatever works for you. But this is one of those structures that most of the students have told me that they were initially afraid that there's a lot going on, but once they drew it, they realized that it really isn't that bad at all. So what we'll end up with is looking something like this. This is from our morning session, and we will actually annotate it a little bit, so there's going to be more information. But we're going to do it from the get-go all together, so I'm not saying that you have to do it by yourself. Anything that I ask you to do, I'm willing to do myself as well. So let's do a little bit of notes together. So what we're going to do, we're going to draw a nephron. And as you see, us guys, we can't talk and do other things at the same time. So I have to kind of focus on one or another. But it doesn't mean that we lost the microphone. I haven't disappeared anywhere. I'm simply trying to do my best. So we're going to be talking about the nephron. And like I said to you, nephron is both the structural but also functional unit of a kidney or unit of the urinary system at large. So we will be focusing on the nephron this time. And there's going to be about 0.8 to 1.5 million nephrons in a single kidney. So there's a lot of those. So we're looking at something that's very, very small, but very important. And because this is so small and it's something that we can't really see with a naked eye, it might seem a little confusing or a little abstract. So for that reason, I want to take time for us to go through it properly. So what we're going to do, before we start looking at the nephrons, we're going to start by looking at the blood supply that's coming into the nephron. So there's going to be blood vessel coming in. And as you see, sometimes my lines are a little wiggly, but that's supposed to be a blood vessel. And there's going to be another blood vessel that's leaving the nephron. And remember, there's going to be lots and lots and lots of nephrons in a single kidney. And I'm drawing this blood vessel that leaves the nephron as well. Here, I'm leaving a little gap. And why I'm leaving that gap, that will become a little more obvious in a bit. But let's give names to these blood vessels coming in and leaving. Well, we are going to be having an afferent, which always meant that it's coming in. And that's going to be an afferent blood vessel, and it's going to be a specific kind of blood vessel. So it's going to be the tiniest blood vessel that we had before the capillaries. So that was, of course, our arterioles. So what we see here is going to be afferent arteriole. And we're going to have then efferent, which was the one that was leaving. Efferent arteriole here. And of course, these can be in any angle or any position, but on this diagram, if it helps you to think about it, you can think of the blood coming in this direction. This is how blood's coming in, and blood leaving to this direction. So that's why we have afferent and efferent. And the other thing, because my drawing is not always the prettiest when I do it on a computer, the other thing that I'm going to do, I'm going to add in a little bit of color, just color in our afferent arteriole here. So this is the tiniest blood vessel before the capillary that's coming in. And then we're going to have efferent arteriole, which was the tiniest blood vessel right before the capillaries, that's right after the capillaries, that's leaving this site. So those are something that we can kind of work out and is partially familiar as a concept for us. Well, what we're going to have between the afferent and efferent arteriole is a bunch of capillaries. And you remember that I used the term capillary network when we were talking about capillaries earlier on. And that simply explained that there was a bunch of these capillaries. So you can think of it as a bundle that's full of these tiniest blood vessel types that we had. And these tiniest blood vessels had a very thin wall, so materials could easily cross out and come back in to these capillaries. So that's what I'm symbolizing there by making it look a little bit like a Swiss cheese. That's the only thing that I'm going to try to show up with that, is that that's a bunch of capillaries. And I'm going to color that in as well to make it a little clearer. And for the sake of kind of being clear, I'm going to add a label for it. So this bunch of capillaries it has a special name. This bunch of capillaries is known as glomerulus. So I'm going to add another arrow there, just pointing to it. So, of course, the glomerulus, now we can work out that that's going to be a site where it's really easy for elements to leave the blood vessels and some elements to come back. But mainly we're looking now of leaving the blood vessel. Well, this glomerulus is actually surrounded. If you think that this is the glomerulus, the network of capillaries, it's surrounded by a structure that circles it from everywhere else but the very top. So on this diagram, I'm drawing a cross-section of this structure that circles the glomerulus. So when we cut it in half, we're seeing that it surrounds this structure completely, with the exception of the very top where it doesn't completely close. And I'm going to use a color to color it in, just to make it a little easier to see where it starts and where it ends. So that's that orangey structure that we see there. And I think that the good thing for us to do would be to label that as well. So let's label that as a glomerular or sometimes the other term that's being used is Bowman's capsule. So it's a capsule around the glomerulus. That's where the name comes from. So either glomerular capsule, or sometimes we also see being used the Bowman's capsule after the person who first identified it. Well, remember that the glomerular Bowman's capsule surrounds the glomerulus from all directions. We're just cutting it open from the middle so we can see what's inside. Well, from the glomerular capsule, we've got a tube leaving, and this tube is a kind of a little wiggly. It can take many shapes. I'm just drawing it fairly simple for us on this diagram, so we don't have to think about it too much. It leaves the glomerular capsule. It could be in reality much longer than what we're seeing here, but we're simply making a diagram that highlights the key points. It doesn't have to be perfectly correct in every proportion, but we see that we've got this tube leaving the glomerular capsule. And this tube can turn around and come back. It can be kind of a wiggly in shape. And this tube actually has a specific name that we're going to use to describe it. This name kind of tells, in my view, a little bit what it looks like. So that's going to be a convoluted tubule. And what we will end up finding is that there's going to be another convoluted tubule in another part of the nephron. So that we don't get confused between these two different convoluted tubules, because we will see another one later on, we have named them differently. This first one is actually going to be proximal convoluted tubule. And you might remember that proximal meant one that's closer to the start of a structure. So this is closer to where the glomerular capsule is at. So that's why it's proximal convoluted tube. Well, after we have had this proximal convoluted tubule, the tube that continues from here actually gets a little bit more narrow. So it's going to be a thinner tube that we're going to see here. And it loops from down and then back up again. So let's draw that tube here. And like I said, it's not quite as thick as the tube that we saw before. So let's see if we can make it here. It's thinner in diameter. And again, I'm going to color it in, hoping that it makes it a little easier to tell it apart from all of these lines that I was adding there. I have a bad tendency of drawing many lines when I'm trying to make something very clear. So it's a little thinner in structure. And this tube is known as nephron loop. Another name that you can sometimes see used about it is a loop of Henle, again named after a person who first identified it. So both of those names would be perfectly good. Nephron loop or loop of Henle. Both describe that structure really well. So it's this thinner tube after the proximal convoluted tube. So it's not probably going to be a surprise to you what the last part of this nephron is going to be called. It's again another wiggly line. Just like the first one that we saw immediately after the glomerular capsule. About the same thickness as the first tube that left the glomerular capsule. So it's again a little thicker. And this is going to be known as our convoluted tubule. I'm going to have to move a little bit of the chat so I can see it. And because it's further away than the first one that we saw, it's going to be distal convoluted tubule. Ooh, I don't know why it sometimes does these long lines that shoot out from what I'm trying to do. So let's do that again. There was no reason for that. So distal convoluted tubule. So distal convoluted tubule is this tube that we drew after the nephron loop or loop of Henle, if you prefer to use that name. And again, I'm just going to color that just simply because I think it's going to be easier to follow this structure once it has a little bit of color. I at least tend to learn by colors and I'm a very visual learner, so I'm just trying to show another way of learning that I would use, but you don't have to if this doesn't feel like your way of doing things. I will not be offended if you don't follow the same technique. So far we've seen an afferent arteriole coming in and we've seen efferent arteriole leaving. So let's just pick those up. Here, the afferent arteriole coming in and efferent arteriole leaving the nephron. Between them, we had the glomerulus, which was this capillary bed. And then we had this glomerular capsule or Bowman's capsule that surrounded the glomerulus. Leaving from the glomerular capsule, we had proximal convoluted tubule, so this thicker wiggly tube coming out that then changed into a thinner tube that shooted down and back up. And this part was known as nephron loop or loop of Henle. And it can actually be much longer than what we see on this diagram. So I've just simplified it a little bit for us. And at the very end, we have another thicker convoluted tubule, and this was going to be the distal convoluted tubule. Well, distal convoluted tubule opens up to somewhere. Let's add that there. Distal convoluted tubule opens up to this duct that we can see here. And in fact, this duct receives secretions from multiple nephrons. So we're going to call that collecting duct. So collecting ducts collect the contents that leave a nephron, and then they carry it to the middle of the kidney where we saw the adaptation of these funnel-shaped structure that collected everything leaving from these tiny structures. So let's label this, and we're going to label this as a collecting duct. And for the sake of consistency, I'm gonna color that as well. But you'll see it has multiple branches, so it's receiving material from multiple nephrons, not just from this one nephron. Collecting ducts really receive a lot of material from multiple nephrons. And then all this material is gathered together and it makes this content that's left from the collecting duct at the very end. So let's just color that in. And there we have the major parts of the nephron and the blood vessels coming in and leaving. But you notice that I left this efferent arteriole kind of unfinished, and there was a reason why I did that. So let's add rest of this efferent arteriole to this diagram and see where it's going to go. So this efferent arteriole actually wraps around and remember this arteriole is very thin-walled blood vessel and it travels right next to the nephron, following its path, also the nephron loop, or loop of Henle if you prefer to use that term. And then eventually joins back to the efferent arteriole that we drew earlier. So it's just this blood vessel that follows next to the other parts of the nephron that we just drew. So this is why I'm using on my diagram color, because I just thought that it might get otherwise a little messy, but I'm sure that we've got better artists, better people at drawing than I am. So your diagram might be much clearer than what I'm doing. And what I will do at the end, I have actually already done that for the morning session. I will upload this to our Connect, our Canvas learning module. So what I'm going to do, I'm just going to take a color and color in the rest of this efferent arteriole. We see that it loops next to the nephron, travels there where the nephron goes, kind of close to it, next to it, all the way to the bottom of the nephron loop or loop of Henle, if that's the term you prefer, and then connects to the rest of the efferent arteriole that we talked about earlier. So here, I think that diagram is taking nice shape now. So these are all the structures of a nephron and the blood vessel that goes into there that we need to know. So you'll notice that there is this capillary that kind of wraps around the nephron, and that's going to be something that's going to be helpful for us. I'm just simply, for the sake of making this a little prettier, going to color in also the heading. So this is our nephron. So when it's this orangey color, that's the nephron part. So now we have a very nice diagram here. These are all the anatomical structures of a nephron that you need to know. This is all the parts that you need to be familiar with. But some of my students actually prefer that they add the next information that I'm going to talk about with you. They add it on a separate drawing, but if you feel that you have room in this drawing, we can add it also on this. But let's go through first all these parts that we have identified here by using a diagram that I drew before. And you don't have to worry about the color change. The color really doesn't matter on this case. I just added it because I wanted to highlight how they're different parts. So we see the efferent arteriole coming in, and efferent arteriole leaving from around the neck. So afferent was like arriving, that's my mnemonic, and efferent was like exiting. And the blood flows in this direction, coming in from the afferent and leaving to the direction of the efferent. Then we also saw the glomerulus, which was the capillary network between the afferent and efferent. And we also saw the Bowman's capsule or glomerular capsule. That was this first part of the nephron that we looked at that circled around the glomerulus. So that was our first part of the nephron. Then we are not going to worry about this arrow, but what we're going to worry about or look at is the proximal convoluted tubule that comes out from our glomerular capsule. After that, we have the nephron loop or loop of Henle, if you prefer. I'm just going to add a charger, connect that so that I'm not going to run out of battery in the middle of our session. And after the nephron loop or the loop of Henle, we had, of course, the distal convoluted tubule, the part that left the nephron, the last part of the nephron. And after distal convoluted tubule, we are going to have our collecting duct. And collecting duct receives secretions from multiple nephrons. And then we saw this loop that went around this capillary from the afferent to efferent arteriole. You saw that I had a little bit of already added some arrows to this diagram, and that's what we're going to look at next. These arrows are going to tell us about the physiology that's involved with the nephron. So really, rather than showing you this diagram, I just drew it with you. There's nothing too much of a surprising about that. So we're going to go back to our drawing here, and we're going to add the arrows. These arrows are going to be, then again, the physiology of the nephron. What really happens? Why did we have to learn about all this? And the first arrow that we're going to add to this diagram is going to be between the glomerulus and glomerular capsule. So some of the material leaves the glomerulus, so from the blood that comes into the glomerulus, and it enters to the nephron through the glomerular capsule, or the other term that we could see being used about it was the Bowman's capsule. So some of the elements from the blood are being picked away here, and they enter to the nephron. And this stage is known as filtration. So during filtration, we take certain elements from the blood that passes through this glomerulus, and those elements enter to the glomerular capsule. So that's the only thing that happens there, and that process was known as filtration. So we're filtering the blood, and whatever we take away from there, from the blood, that then is gonna enter to the rest of the nephron. Well, then when we get to the end of the proximal convoluted tubule and we get to the nephron loop, or the other term that we could see for that was the loop of Henle, there's going to be another arrow, but this time this arrow is going to be pointing out from the nephron. So some of the material that we've taken into the nephron gets put back into a blood vessel that's running next to the nephron. So we took something away from the blood, but then part of it we returned back to the blood. And this stage is known as reabsorption. So during reabsorption, we actually return back some of the material from nephron and put it back to the cardiovascular system, to the blood vessel running right next to the nephron. That was our reabsorption. Well, we're going to have two more stages that take place in this nephron. Well, then when we go to the further end, the distal end of the nephron loop, or the loop of Henle, and at the beginning of the distal convoluted tubule, there's going to be another arrow, but this time it's going to be pointing from the blood vessel back into the nephron. So material gets put back in to this nephron. So we're taking material from the blood vessel and we're putting it to the nephron at this point. And this event is known as secretion. So during secretion, material moves to the nephron from where we would have the blood vessel running next to the nephron from this capillary. It gets put back in to the nephron. And we only have one more arrow to add into our diagram. And this final arrow is going to be here, where from the distal convoluted tubule, the contents are being dumped Oh, I'm going to redraw that. That turned out a little ugly. So from the distal convoluted tubule, the contents are going to move on to the collecting duct. So this would be essentially what is being shown here. This arrow are urine, the pee that your nephron has made. So the final product of all these stages is shown here. And this is known as our excretion. So as you'll notice with the nephron, there's multiple steps. So we can ensure that what we want to take out from the blood gets taken out, and what we want to maintain in blood still remains in there. So that's why we've got almost this like over-engineered structure with multiple steps of ensuring that we're doing the exact right thing. Right things get taken away and right things remain in the blood. So that's the function of these multiple physiological steps. And that's what you need to know about these physiological steps. And I'm going to again go back to where we were earlier on just to show that with the arrows that I have included on this diagram. So just like we saw before, let me take my laser pointer from our glomerulus, which was this network of capillaries, material moves to our glomerular capsule or Bowman's capsule. That movement was known as filtration. Well, from the proximal convoluted tubule and the nephron loop or loop of Henle, material moves to the blood vessel that runs next to it. This was known as reabsorption. Well, from the blood vessel that's running next to it, material moves to the nephron loop or loop of Henle and to the distal convoluted tubule. And this movement is known as secretion. And at the very end, whatever is left and leaves the distal convoluted tubule to the collecting duct, that's known as our excretion. So we did a pretty good job drawing that diagram ourselves, I'd say. So I'm very, very proud if you followed with me on that. And this is really the major part of this chapter. I'm just going to show a couple of other things. One thing that I want to mention is that we sometimes see in medical notes the GFR. GFR stands for glomerular filtration rate. So what glomerular filtration does, it measures how much blood passes through the glomeruli, which we saw here, in each minute. So essentially what that number, which is a number that we see in lab reports, it tells us is that how well your kidneys work. And we sometimes need to have that knowledge, not just because we want to check that your kidney is working, but if we're going to give you a drug or some sort of a contrast agent that gets cleared off from your body. So then we want to make sure that your kidneys work well enough to clear it off from your body so it doesn't stay there forever. So that's why the GFR, glomerular filtration rate, is an important lab number that we sometimes check. The other thing that I wanted to discuss was a hormone that you've seen before. It was abbreviated as ADH. This hormone is, of course, our antidiuretic hormone. And you remember that we talked about it on the chapter of endocrine system at the very beginning of this semester. But now you actually see what the antidiuretic hormone does. So the name tells us that it makes us less urinate than what we would normally do. And you'll notice where it works on the distal convoluted tubule of the nephron. So antidiuretic hormone turns the distal convoluted tubule of the nephron and makes it very permeable so that the materials can leave out of this part of the nephron and go back to the blood circulation. And if materials are leaving, well, then we're going to make less pee. So it's anti-urination, anti-diuretic. So it's anti-diuretic hormone. It stops us from making as much pee. And the way it worked was through the distal convoluted tubules wall becoming more permeable. And that really completed the key parts of this chapter. I'm quickly going to mention about the ureter, bladder, and urethra. But before I do, I just wanted to mention regarding the urine. So you'll notice most of the urine is water. So that's why urine is a good way of manipulating that whether we have a lot of blood or less blood in the system. So we're able to change the blood volume, so blood pressure through changing the volume by manipulating how much urine we make. And majority of the urine was water. About 5% of it is solutes. There's various solutes that are waste products, such as our urea, uric acid, and creatine. We need to get rid of them from the body. And the way we're getting rid of them is by peeing them out. There are other solutes that we normally can find as well, and I've listed them there. But what's really important for us to know about the urine is what we don't want to find there, what we should not find there. And what we should never find in urine is blood. So if there's blood in the urine, something's wrong. That's a cause for concern. The other thing that we do not want to find in the urine, in addition to the blood on its own, but we also don't want to find the white blood cells. And you learned in the previous chapter that the white blood cells were an indication that there's an infection going on somewhere in the body. So that's not a good sign either. But we really do not want to see blood in the urine. And now I'm just quickly going to go through the ureters, urinary bladder, and urethra. And as we have two kidneys, we're going to have two ureters. So ureters are these structures that connect the kidneys to the urinary bladder. And they are muscular tubes that have lines that are lined with smooth muscle lining. And that's important, this smooth muscle. Well, the smooth muscle lining of the ureters is important because it is thanks to this smooth muscle that it's able to kind of squeeze and move the urine to the direction that we want to move the urine. So even if you were standing on your head, like this lady on this diagram or this slide of mine, still the ureters would be carrying the urine from the kidneys to the urinary bladder, even if it was against the gravity. And this is how, for example, our astronauts still have perfectly functioning urinary system. Even if you're lying in a weird angle, urine is going to be moving from the kidneys to the urinary bladders, thanks to this smooth muscle lining of the ureters. The next thing that I want to talk about is our urinary bladder. And you'll notice that the urinary bladder receives ureters from both sides, and these ureters enter the urinary bladder actually at the bottom of it. There's another tube leaving from the bottom of it, which is our urethra. And we're going to talk about the urethra in a bit. But if you look, and this is a diagram as if we would have cut open the urinary bladder and we would be looking in there. We will see these two openings for our ureters. So both ureters coming from the kidneys, entering to the urinary bladder at the very bottom of it. And then we have more anteriorly at the front part, we have our urethra, which was the way for pee to leave the body. So because they form this kind of a triangular shape, we describe this area as a trigone. And trigone can be a very useful anatomical landmark. For example, when you put a camera inside the urinary bladder and try to orientate yourself there. Well, one thing that we notice with the urinary bladder is that the urethra plays an important role whether there's infections or not. But before we talk about that, let's remind ourselves that the urethra is this tube that leaves the urinary bladder and carries the urine to the outside world. And there it is a muscular tube, and there's multiple sphincters, so we're controlling that when it is open, when it is not open. Usually we don't have any problems with that. We're fairly well able to control of keeping the urine inside the urinary bladder when we don't want it to leave. But there is a significant difference between the male and female ureters. As you see on this female urethra, the length of the for our female diagram, we have here the urinary bladder. We have here the parts of the ureters coming in. The female urethra is much shorter when we compare it to the male urethra. A female urethra is typically about 4 centimeters in length, whereas the male urethra is somewhere between 17 and 19.5 centimeters in length. Well, because this length is so much shorter, it's much easier for bacteria to climb up and get into the female urinary bladder. So that's why females are more likely to develop urinary tract infections, UTIs, than what males are. And final thing that I want to discuss with you guys is the process of micturition. Micturition is the proper medical term for the process of expelling urine. So process of peeing out the urine. So rather than saying I need to go to the bathroom to pee, you can be very, very fancy and tell that, oh, I need to go and practice some micturition in my privacy. So that's simply the term that I want you to know about. And that really completes the key points that I wanted to discuss with you about the urinary system. What I'm going to do now, I'm going to jump out of this presentation. And bear with me as I navigate without seeing what's going on. And we're going to open our initial presentation just because there's few little final points that I want to discuss with you. Provided that the computer is still going to work with me. It's thinking a lot and what it should be doing next. But I think that we can make it work for the end of this day, even though it's been a long day. So let's get that presentation shared with you. Here we go. So you should be able to see my PowerPoint presentation. And I think that we got here. We talked about the key points of the urinary system and especially about the nephron. The very last things that I want to mention with you guys was the fact that if you did want to take any missing or retake assignments, just send me very clear details that what assignment it was. The Canvas messages are usually the best way to reach me. I also wanted to remind you that you do have tutoring available. The tutoring is still in an online format. So we use the Cranium Cafe, and all you have to do is use the link. That link is posted in many places. So you can then find a tutor who will work with you if you have anything that you want to be explained or go through or any tough questions. Of course, I, as well as your instructor, will always be more than happy to make time with you. I have actually had quite a few requests and quite a few one-on-one virtual meetings recently with the students from this class. So all you have to do is say that you would like to have a meeting with me, and we'll schedule that, and then we can discuss in a virtual format, in live format, whether there's something in terms of the chapter material or about the assignment or anything else that I can help you with. So the final thing that I have to ask is that if you have any questions about this chapter, about the assignments, or anything else that I can help you with. But if you don't have, that's all I had for today. So I open, if you have something to ask, you just let me know. If you don't have anything to ask, that's really all that I had planned for today.
Thank you, Professor.
Thank you. If you're happy and you know everything you need to, I'm super happy, and I will just say good night and thank you for joining me here. If any questions come up, just send me a message.
Okay, that's it. Okay, I'll send you a message if I have any question. Have a good night.
I, I can address it now. I'm going to pause the recording so we can. So we're back just to finish off this recording. And what I actually discussed with the student was week's assignment, where you will be actually doing a clinical case scenario. And I'll give all the information that you need to know about that clinical case scenario, how to answer to that in the next week session. But if you do want to make a head start, the instructions are pretty clearly written there. You have to download a file and then fill it and submit it back to me. But that's all for the next week. We've covered all of this week's material. I think we were pretty successful in tackling everything that I had thought about. And like I said, really the only big thing on this week's material is that remember that the Fills Lab, you might need to do some clicking around to get the Flash player to run, but we should be good. The other question that the student asked from me was that when are the exam 2 and practical 2 results ready? I will try to get them ready tomorrow, but I can't promise that. So I'm actually away on a conference, but as soon as I get them ready, I will send you an announcement to let you know that they are finished and your final grade is the final grade that you're seeing. So bear with me, we'll get there together. And I thank you for joining me tonight and hope you have a very, very good week. Remember, I'm here to help. All you have to do is reach out to me. Let's make it a great week. Bye.